Click for IB Biology. Here we're looking at the virus HIV. There's a PDF, a blank PDF, down in the description, which you can download and annotate with your own notes. Later you can take the quiz to secure your understanding. There's a prompt structure to assist you in delving into greater detail. And then we have links to other videos in the series, to the channel, and to the playlist. HIV infection is dealt with in a number of places on the course, including the immunology part, to which students are referred for further details on the disease itself, particularly the acquired immunodeficiency syndrome aspects of HIV infection. Here we concentrate on the virus cycle. So here we have HIV, a diagram of HIV virus, the immunodeficiency virus, and this is a single-stranded RNA positive sense virus, but the curious feature is that present within the capsid are two strands. These are not forming a double helix. There's ×2 of the strands. Now this is understood to play a role in the correction of any translation errors which occur within the life cycle of the virus. As an enclosed enveloped virus, it has on its surface host proteins, glycoproteins, but also introducing, with the expression of genes in here, spikes have been introduced to the surface of the HIV virus. And these are in two parts. One part is called glycoprotein 120, and the other part is known as glycoprotein 141. So the virus particle comes into contact with the cell surface receptors of CD4 T helper cells. These are central organizing synergistic cells of the immune system. Once more, I refer students to the immunology part of this course, where the helper cell is explained and developed for your purposes. So this is what the HIV virus targets. Now, given that helper cells coordinate the response of the immune system, then the HIV virus is targeting one of the central cells in any immune response. And it will be detected and lock onto with the GP120, locks onto what is called the CD4 receptor. So this is a glycoprotein in the surface membrane of a T helper cell, onto which the enveloped virus attaches. Now, once that's happened, then the second component, the glycoprotein 41, interacts either with a cell membrane structure called CCR5 or CXCR4. And what this does is brings about fusion with the cell membrane. Now, this is not endocytosis, this is fusion. And so we have the incorporation into the cytoplasm of the CD4 T helper cell of the capsid structure of the HIV virus. And then the protein capsid is uncoated to release the single-stranded RNA. Now, as a retrovirus, what then happens is that the RNA is reverse transcriptase enzyme converts this into viral DNA. This is absorbed through the pores of the nucleus into the proximity to the host DNA, where it is integrated into the host DNA and is now known as a provirus. The enzyme which integrates is called integrase. Now at that point, the virus can become dormant for significant periods of time. But as with other lysogenic cycles, it is possible that when triggered, it may enter the lytic sequence, in which it will then bring about the synthesis of the capsids and the RNA, which will then combine together before what's known as a budding process, which releases the viral DNA, the capsid, surrounded by the host envelope, and thereby raising the levels in the bloodstream of HIV, which is called the viral load. Now, of course, this can then lead to the opportunity for reinfection of other CD4 cells, or indeed possible transmission to another person. HIV, like many other viruses, is subject to a high mutation rate, largely because it doesn't have proteins associated with it. Also, it's possible for recombination to occur, creating variations on the original DNA method. As a human disease, its treatment has significantly impacted on people who turn out to be HIV positive. The so-called ART treatment, anti-retroviral treatment, anti-retroviral therapy, targets the idea of suppressing the lytic cycle and therefore reducing the viral load to undetectable levels. So if it's possible to reduce the viral load to undetectable levels, then the possibility of infection is greatly diminished. But remember also that by preventing the budding process and the lytic cycle, this also prevents the damage to the CD4 T helper cell, and therefore prevents any inability to respond to a particular infection. This aspect of the infection is developed in the immunology videos of this course. Appearing before you now are links to the channel, and also to the playlist and to the next video in the sequence, along with the viral playlist.